I am very honored to have these two invited speakers today, Doctors Donald and Ada Olins. Since they are both going to talk in this seminar, I'm going to introduce one first and then the other. Dr. Olins was my thesis advisor. For those of you who don't know that fact, so that's one of the reasons why they're here. So Don got his bachelor's degree from the University of Rochester in New York. He started in the medical school in Albert Einstein, but saw the light and decided to do a PhD in biochemistry. This is the department that I'm also a faculty of here in our school at the Rockefeller University in New York. Then did a postdoc at Dartmouth Medical School. Ada, on the other hand, she did her bachelor's in chemistry in City College of New York, started graduate school in Harvard University, and obviously for obvious reasons, she transferred to New York and did her PhD at NYU in biochemistry, and also did a postdoc in Dartmouth. Both of them then accepted positions at the University of Tennessee, Oak Ridge Graduate School of Biomedical Sciences, where Don, for example, he went through the ranks from assistant to professor, where he retired from Oak Ridge in 1998. I was a research professor at the same institution, and they have been visiting scientists, visiting professors in many institutions, especially in like Germany, in the German Cancer Research Center. After retiring from Oak Ridge, they have continued to do research at the Foundation for Blood Research also at the University of Maine. They're presently faculty associates and members of the graduate faculty. They were visiting scholars, both of them, in Bowdoin College in Maine, in Brunswick, Maine. Visiting scientists also at the Maine Medical Center Research Institute at the present time, and research professors presently at the University of New England College of Pharmacy in Portland, Maine. They have received many, many honors throughout their careers, but if I were to list them, we wouldn't have time for the seminar. I think one of the most well-known accomplishments of the Olins are their description or the publication of the first micrographs establishing the structure called the nucleosome, the chromatin subunit, and also the description of its speculations on its composition, the composition of the nucleosome, and its relationship to higher levels of chromatin. And in fact, that publication in Science in 1974, in 1983, was already considered a citation classic, which, for those of you who remember current contents, that used to be the citation classics were used to present the human side of scientific papers, especially those that were highly cited. So only those highly cited publications were chosen for citation classics, where the investigators would then describe how they came about the discoveries they describe in their highly cited publication. So in 1983, that science paper in 74 describing the new bodies, or the nucleosomes as they're known now, had been cited already over 790 times in 1983. So I don't know what's the number right now, but it should be pretty high, much higher than 790. So we're going to have then Drs. Ada and Don Olins give us a seminar. The title is going to be Nucleophilia, or the Love of Nuclear Structure. We're going to start. Thank you very much. Yeah, we can hold it.
Thank you very much, Carmen. It's really a pleasure to visit you and to see this big department and big graduate program, and to remember from the days when you were a student to see now with your professional success. It's quite a thrill for us. So thank you for your introduction. I'm starting out with my picture of a lighthouse, which is very close to where we live in Portland, Maine. But do I have a pointer? This?
Yes.
And this.
There, right.
Thank this is something many of you don't recognize. You might think it's salt or sugar, but no, that's snow. And in Maine, we have quite a lot of snow. The way we've divided our talk, we always like to both give parts of the talk because we work together. Is the sound okay?
Yes.
So I will talk about lamin B receptor and the control of nuclear shape. And as an example, I'll talk quite a bit about granulocytes, neutrophils, which are white blood cells which have a polymorph nuclei, and talk about the shape and what determines that. Don is going to talk about something quite different, and that is the heterochromatin near the nuclear envelope. We've named it epichromatin, and he'll talk about that and the role of phosphatidylserine in epichromatin. So this is a review to all of you, I'm sure. I just wanted to point out several parts of the nucleus. You'll see that I'm quite interested in microscopy, and I'll emphasize microscopy as a method for doing research. The nucleus has around its periphery heterochromatin, these darker regions interrupted by nuclear pores which allow the transfer of materials in and out of the nucleus. There's also heterochromatin around the nucleolus, and in the cytoplasm, mitochondria here, and endoplasmic reticulum. I want to point out that the nuclear envelope is a part of the endoplasmic reticulum. And I'll show that in a little bit higher magnification. So the ribosomes on the surface of the nuclei are really on the surface of the endoplasmic reticulum. Here's a higher magnification electron micrograph, and just so you can see the nuclear envelope, there's an outer nuclear membrane, perinuclear space, and then an inner nuclear membrane. Here's a nuclear pore, and then the heterochromatin. I want to point out that along the surface of the inner nuclear membrane, the chromatin is arranged in a little bit more uniform, a little bit more condensed pattern than in the inside of the nucleus. So here a cartoon of this area. This is supposed to be a nuclear pore. The ribosomes on the endoplasmic reticulum, on the outer nuclear membrane. And then there are many, many proteins which are a part of the inner nuclear membrane. They're supposed to be something like 60 or 70 different proteins. We're going to primarily concentrate on lamin B receptor because it plays a role in the shape of the nucleus. We've put a couple of other jelly beans of proteins that play a role in the inner nuclear membrane that have been studied extensively. There are many that have not been studied yet. But there's this layer of proteins which are a part of the membrane. Then there's a region of a layer of lamins. The lamins are not really inserted in the nuclear envelope, but form a layer just under it and have a role in the stability and shape of the nucleus. And many of these things contact the chromatin, HP1 alpha, lamin B receptor, and it's a complicated system which we're not going to be able to discuss at length, but lamin B receptor is the one to concentrate on because that's a very interesting molecule. So in order to study the shape of the nucleus, we decided to study granulocytes. This is a neutrophil, a granulocyte, in the bloodstream. You see the red blood cells around it. And what is it doing? It's doing something very important. It's chasing a bacterium. These cells are our first line of defense in fighting bacterial infections. So they not only have to move very rapidly. Did it stop? Oh, I was going to let you see it again, but we're having it not only moves very rapidly, but it also, these cells have to squeeze through very narrow spaces. the thing I'd like to emphasize is that these cells have to be very flexible, very pliable, and the most rigid part of the cell has been thought to be the nucleus when it's a round, ovoid, very firm structure. But these cells have a nucleus which is much more flexible. That's seen in this electron micrograph, which came out of the Dvorak lab, and you can see the this is a layer of endothelium. The blood is on the upper left-hand corner, and you see two granulocytes waiting to pass through the endothelium. And here, beautiful image of the cell having just passed through, and part of it is still in the inside of the blood vessel, and most of the cell has squeezed out. So it's thought that the reason these have lobulated nuclei is that it gives it flexibility and allows the cells to squeeze through very narrow spaces. These spaces might be as narrow as 1 micron, where the nucleus is more than 10 microns, so it really has to squeeze. Now to introduce you to granulocytes, both in human and in mouse blood. Here are several examples of the lobulated structure of the nuclei. In mouse, it's even more interesting. The nuclei are lobulated, but also a toroidal shape. And these little structures are heterochromatic regions. In mouse, heterochromatin pericentromeric chromatin is very rich in AT, and so it forms these chromocenters which stick out like drumsticks. In humans, we sometimes see the drumstick, and been shown that those contain the inactive X. So these are regions of chromatin which are highly compacted and seem to be able to change the surface of the nuclear shape. So now I'm going to talk about lamin B receptor, which is the protein that we have found which is involved in determining the shape of nuclei. We're going to talk about mutations both in human and mouse diseases and what that does to the shape of the nucleus. Here are some blood smears, human blood smears, and there is an anomaly called Pelger-Hewitt anomaly. And in the heterozygote, the nuclei are bilobed. In the homozygote, the nuclei are ovoid. Now, this is a very rare occurrence, because if there's absolutely no lamin B receptor, it seems to be a lethal mutation, and there is something called Greenberg's dysplasia, where fetus dies in utero with lamin B receptor mutations. We had the good fortune of working with colleagues in Berlin, Carl Sperling and Katrin Hoffmann, who studied the mutations in people with Pelger-Hewitt anomaly. It turns out that there's a town in East Germany that has a very high incidence of Pelger-Hewitt, and they did a genetic study. And when our paths crossed, we had a very interesting time. Katrin had just mapped the mutation in this community and said, look here, it's on chromosome 1. These are 100 genes that could be in that region. What do you think it is? And Don looked at it and went down the list, and lamin B receptor, which we had been studying already in a tissue culture line, jumped out at him. And indeed, to make a long story short, when it was tested, lamin B receptor was the mutation in this community. We had the good fortune, Katrin is a physician, so she was able to make contact with a family and allowed us to study the blood of a family. The parents were heterozygotes, and the young man was homozygote. And these are some of the problems that he had. The nuclei in his granulocytes were ovoid. There was some mental retardation, disproportionate body, macrocephalus. We don't know whether this heart defect had any connection to the mutation or not. And the skeletal short metacarpals were also seen. When looking at the blood of this family, this is a wild-type human granulocyte. This is a thin section electron micrograph, so the lobulated nucleus looks like 4 little nuclei, but in fact that's all one nucleus cut in that plane of the cell. The homozygote, you see, is quite different. The nucleus is ovoid and the chromatin is redistributed. Quite different from the wild type. In mice, this is another story which is interesting, that in Bar Harbor at the Jackson lab, Leonard Schultz had been studying mice that had a skin problem. Ichthyosis was the mutation, and they had reported that the nuclei in the blood of these animals was quite different. And again, to shorten the story, these mice have a mutation in lamin B receptor. And let me show you what the blood cells look like. Here are 2 wild types. Excuse me, this is the wild type, 2 heterozygotes, and the homozygote. And it looks very much like the human Pelger-Hewitt, and indeed the mutation is also in lamin B receptor. Here are the electron micrographs. You see that the heterozygote is not really bilobed, but it does, it still has the annular shape of the nucleus, but in the homozygote, that's quite different. And again, notice that the heterochromatin is redistributed in that cell. So let me tell you about lamin B receptor, because biochemically this is a fascinating molecule. It's kind of a schizophrenic molecule. The amino terminus, the first 208 amino acids, which are point into the nucleus. Here's the inner nuclear membrane, outer nuclear membrane. So this amino terminus is facing into the nucleus. There's a globular region. This has been shown to be a Tudor domain, a hinge region, and a globular region. The things that bind in this region are lamin B, the HP1 proteins, heterochromatin proteins, and the chromatin binds in this region. Then there is a region of 8 presumptive transmembrane domains, and that part of the molecule has a sterol reductase activity. So we have 2 very different kinds of functions. This is very important in cholesterol biosynthesis, and so we don't really know why these 2 parts of the molecule have been joined in evolution, but that's something very interesting to consider. This is another diagrammatic showing you the Tudor domain here binding to chromatin, then the region that binds to the lamina. This little meshwork is a fanciful version of the lamins, and then the HP1 again binding onto the chromatin, and then this sterol reductase region which is nestled in the inner nuclear membrane. For those of you who really like the chemistry part, I want to show you where in the pathway of cholesterol biosynthesis, here starting with lanosterol, the lamin B receptor plays a role. And it has been shown that this is indeed an important part of the function of lamin B receptor, that it does participate in cholesterol biosynthesis. And the homozygote in the of mutations in the region that has a sterol reductase activity, those are the fetuses that die in utero in the Greenberg's dysplasia. disease. There are other things in the cholesterol biosynthesis, other steps which have important biological functions. I won't get into it, but some of the enzymes involved there have a lot of homology to the lamin B receptor. So I want to get into two cell systems that we've worked with that have allowed us to analyze some of the problems in nuclear shape. One of them is a tissue culture line, which is called HL60. We use a particular subgroup, which is S4, which goes through development a little more rapidly, and in 4 days rather than 6 days. But HL60 is an AML line that's been used in laboratories for many, many years. The nice thing about this cell line is that it can be differentiated in culture, and I'll show you that in a minute. EPRO cells are mouse cells, which are similar. I can't get into the whole way these were raised. These were developed by Peter Gaines, and we have a line that has the lamin B receptor mutations, and we could study those also in mouse cells that can be differentiated in tissue culture. So here the HL60 line, the control group, or the group that's not been treated chemically in any way, just continue to grow. They're very robust. They grow in solution, and we have them in our lab even now. If you treat those with retinoic acid, you get a granulocytic form of the cell, and these live for 4 or 5 days. You see the nuclei become highly lobulated, and then they go into apoptosis. If you treat them with 4-bol ester, they develop into macrophage, which have an ovoid nucleus. And if you treat with vitamin D, they become monocytes, which also have ovoid nuclei. When you treat them with retinoic acid, RA, we see the ovoid nuclei in control, and then the differentiation, we get lobulation. This is very low magnification, so you can hardly see it, but here at higher magnification, there are 4 big lobules in this section, and you see other structures, and these are structures that we've been very interested in because of the kind of chromatin conformation that we see in these. Let me quickly take you through this. On the bottom left here, you can see a portion of a nucleus with a nuclear pore, the very regular chromatin right under the inner nuclear membrane that I mentioned earlier. Then let's go out here. This is cytoplasm. Then another outer nuclear membrane, inner nuclear membrane, and then a layer of chromatin, and then inner nuclear membrane and outer nuclear membrane. So imagine these things. These are not just strips. This is not a sausage. This is a sheet, because we've got a thin section cutting through the cell, and these are sheets of chromatin, and they're very uniform in diameter. And I think this reflects a conformation of chromatin that's very important. One of the questions in chromatin structure is, what is the level of organization above the nucleosome? And whether there is a 30 nanometer fiber or not, whether this is helical, uniform, there have been many controversial structures. But here's a structure that's approximately 30 nanometers in diameter, and it is very regular. We've done a fair bit of biochemistry on the proteins during differentiation, and this is a little bit complicated, so let me take you through it. On the bottom is a Kamasi blue stain, just so you can see that the loads are pretty uniform going across the gel. The end lanes, U and A, are control cells. This is an A431 cell line, and this is U2OS. And it's just to show you that the conditions are good to demonstrate the different antibodies that we reacted with on a Western. Now, 0 is untreated HL60 cells, and then going to the left with R are retinoic acid treated cells. going to the right with a T are TPA, or the four-bol ester treated cells. So to the right is macrophage. Remember the ovoid nucleus. To the left is the granulocytic differentiation of the granulocyte, of the lobulated nucleus. Very interesting is that lamin AC is very low in the granulocytic form. It goes way up in the macrophage. Lamin B1, not so clear, maybe a little bit of a reduction, but not dramatic like the lamin A. Lamin B2, I would say that's unchanged. It's not beautiful. Lamin B receptor is very interesting. As you go to the granulocytic stage, we have a very dramatic increase of LBR. Lap2 beta is fairly uniform across. Emerin also not much change. We have a cell line that was developed by Orly Ernst in our lab, and actually we worked with her in Germany. And the S4 line, now treated with retinoic acid, you've already seen this, the ovoid nucleus going to a lobulated nucleus. The knockdown of LBR, which we call SH1, is ovoid nuclei, both with and without retinoic acid. So we have no lamin B receptor and no lobulation. Here is some of the biochemistry on those knockdown cells. Again, SH1 is LBR knockdown, S4 is our normal cell line. Lamin B receptor, of course, doesn't go up in the knockdown cell. It's not working all the time. Laminate, the ones where we don't see a change, we've not put a star next to the lane, and lamin AC with retinoic acid drops in both lines. We have a much lower starting concentration. Sun1 likewise drops with retinoic acid. Vimentin, which is a cytoplasmic protein, also drops. Here again is the, well, it's Ponceau S, just like Kamasi Blue, to show the loads on the gel. Now I'm switching to the EPRO cells. These are the mouse cells that we have the LBR knockdown. And just to show you what happens to the nuclei in those cells, there are essentially three lanes here: the wild type, LBR expressed normally, the heterozygote, and the homozygote LBR knockdown. You see that when you treat with retinoic acid, this is the medical abbreviation for retinoic acid, ACRA, the same material. When you treat with retinoic acid, you get the annular shape and a pretty big increase in the amount of LBR. In the heterozygote, you still get the annular shape, and you get some increase in LBR with the mutation, the IC or the knockdown. There is no LBR, or very, very little. This is lamin B1, just to show you the changes in the nuclear shape. These are all the same field going vertically, so the same cells, and here the DAPIs. So the one thing I want to point out here is that the condensed chromatin, these chromocenters which stain very brightly with DAPI, come to the center of the nucleus, as opposed to here in the wild type where it might be on the edge or anywhere. Here is another demonstration of where the heterochromatin is in these same cells. This is organized exactly the same way as the previous slides. So we have the wild type, the heterozygote, and the IC cells. And you can see the heterochromatin. These are confocal sections, so you can see the distribution more. It's just a single slice. But the thing I want to point out, that in the IC mouse, the nuclei are ovoid and the chromatin is totally redistributed, as seen by heterochromatin markers. The markers we used in this case was HP1 gamma and trimethyl H4K20. These are established markers for heterochromatin. And as I pointed out earlier, in mouse it's really nice because you can see the DAPI localization so clearly. There are several other things that we've found that play a role in determining the ability of lobulation in a cell. Microtubules are required to have lobulation, and I'll just show you one data point where we studied this. This is a special line of HL60 cells which live longer than 4 or 5 days. And look at the coding here. Ovoid nuclei are the red ones, and then slight indications of indentation or lobulation or multilobe. The first thing you see in the control, where we do not use any no-cortisol, just treat with retinoic acid, the ovoid nuclei decrease and the lobulation increases. If you add no-cortisol, which destroys the microtubules, then you find that the nuclei do not lobulate, and you have an ovoid nucleus regardless of the no-cortisol concentration, 0.1 or 1 micromolar no-cortisol. So bear in mind that's another thing that you need to have lobulation is a good microtubular structure. And another thing that is important in the granulocyte nuclear shape is the link complex. And the link complex are those proteins which link the nuclear envelope to the plasma membrane in the cell. And I will just show you a little bit of data here, a cartoon of link proteins attaching to actin filaments, to intermediate filaments. These proteins are called plectin, nesprin, the sun proteins, and they are a part of the system that gives the cell flexibility. And let's see what happens to those when you differentiate. By now you're familiar to our way of arranging our Western blots. TPA differentiation to the macrophage on the right, 5 days of retinoic acid differentiation on the left, and again the control A and U2OS cells and A431 cells. Now we're looking not at nuclear proteins, but at cytoplasmic proteins, the ones that are members of the link family. And notice that vimentin really decreases in the granulocytic type differentiation, and Sun1 is gone in the differentiation in both directions. So we want to say that it looks like these factors, the link proteins, which are involved in giving the cell rigidity, are decreased in cells that have a lobulated nucleus. So I will just summarize briefly, and I hope I've left Don enough time for his story. Neutrophil nucleus shape and heterochromatin distribution appear to be a consequence of sufficient levels of LBR, of reduced laminase C, and the presence of intact microtubules. LBR is an essential enzyme in cholesterol biosynthesis. I think medically this is something that still needs to be explored in greater detail. The other thing that we have seen is that lamin B receptor seems to increase the synthesis of membranes, and that exemplified by the ELKs that I showed you, those sandwiches of chromatin. Those seem to be in part a consequence of an increased synthesis of nuclear envelope. And then granulocytes are very pliable cells. I think I've emphasized that a lot, and their flexibility is probably due to a reduction in the link complex proteins. So with that, I build a nucleosomal bridge for the connection between our two talks. Here's Brahma. Right there.
Well, first of all, I want to express my appreciation to Carmen for bringing us here and for arranging this terrific visit. We've met only a few of your colleagues, but we're very impressed with the quality of the scientists that we've met so far. And we also, just a few days ago, got questions from the students in the graduate program. We thought we were all finished with our work in preparation for the seminars, and then this letter came with 5 pages of questions by the students. And we've been working our can off to try and think about some of these things, but we were very impressed with the quality of the questions that the students came up with. So we commend you for having such a fine program here. We really are very inspired to see this. So Ada has had the difficult chore of summarizing our 12 years of work after we moved to Maine from 1998 to about 2010. And I have the daunting task of summarizing 1 year of work. Maybe 2 years of work, if you wish. But fortunately, she built the necessary information for me. Okay, so I want to talk about this entire talk that I give is going to be focused pretty much on that thin layer of heterochromatin that's right underneath the nuclear envelope in an interphase nucleus, which we think must be an exceptionally interesting bit of chromatin. So the nuclear envelope associated heterochromatin. And I am afraid I have to go back and review a little bit of what Ada has done. We think it's so interesting that you're going to have to hear it several times. Okay. These are the ELKs, envelope-limited chromatin sheets. We did not discover those. They were discovered by someone that we worked with in he discovered them in the early 70s, the late 60s, early 70s, and we went to work with him in London, 1970-71. These are the ELKs, and these are 2 sheets. And you can see again, I'll just follow that path. This is cytoplasm. This is the outer nuclear membrane that was studied with some ribosomes. Then the inner nuclear membrane, you can see that a little bit better in these images than what I just showed you because they're bigger. Then comes chromatin, and then inner nuclear membrane, the cisternae between the inner and outer nuclear membrane. And then the outer nuclear membrane, you're back in cytoplasm. Same story here. But the important thing is to see how regular this is, how uniform this is. And there'll be a number of pictures like that. Okay. So now comes a confocal movie. And am I glad we got these movies working. You don't know the trauma we went through. We're PC people. And when we tried to do this on a Mac, forget it. It was awful. And, it'd be like going into, some favorite movie you want to see, and they display it at the movie house, and there's no movement. And that's what anyway, so this is the HL60 cell. It's been treated with retinoic acid. One of the things that happens, the red is staining microtubules. And one of the interesting things is that the cells polarize because they want to start migrating. We don't know which is the head and which is the tail, unfortunately. But this is the centrosomal region down here and polarized microtubules. Anyway, the green is a lamin B staining. You can see the nucleus is lumpy, so it's somewhat lobulated. And this cauliflower-looking structure are the elks, these sheets. And this is a confocal image that we've turned into a movie, so it's just rotating back and forth. Okay. Then this is electron microscope tomography. I don't know how this is going to come. Is it coming? It's coming out all right. Okay. Okay, so now you're going to have to get used to this, and I'll guide you through it. What it does is it goes through a series of slices. It's not rotating like the other one was. You're looking at section after section, and then it jumps back to the beginning. Okay, so this is done in an electron microscope, and then it's by computer. The 3D reconstruction is calculated, and then these are the computer-generated sections. Okay, so what you've got here, this is ELKS here, and it's attached to the nucleus here. This is the attachment. And then this is another sort of micro lobe here, so that the ELKS have split into two sheets. Now, we're going to have to just focus on one area to begin with as it jumps. Okay, what I want you to see is that sometimes you can see some internal structure within within this sheet. So here you can clearly see the inner nuclear membrane and chromatin and another inner nuclear membrane. And you can also see this layer under the inner nuclear membrane here. You can also see here, you can see these, what may be cross sections of some higher order structure. Okay. So again, these are sheets of very highly organized chromatin. And they are extremely highly organized. Sometimes these are actually the BCL2 cells that Ada mentioned in passing that we used for the nicotazole study. And the reason why we had to use BCL2 essentially is anti-apoptotic. So if you were to add nicotazole to the HL60S4 cells, they tend to die from apoptosis. And if you use overexpressors, BCL2 overexpressors, they survive longer. They can survive up to 3 weeks in culture. They're not growing any longer. But in the process, they keep building these bigger and bigger multi-layered structures. And also, the nuclei become more lobulated. You get hyperlobulation. I'm not going to show you any pictures of that. One of the student questions was asking about the hyperlobulation that we get in megaloblastic anemia with folic acid or vitamin B12 deficiency. This might be an interesting model for that because this kind of nucleus is highly multilobulated. And whether that means, how that's interpreted in terms of megaloblastic anemia beats me, but it'd be a fun thing to discuss. Anyway, what you see is these multiple layers that have been separated. So this is a chromatin layer with two inner nuclear membranes, one on either side, then a gap. There's no outer nuclear membrane, but you go right into another inner nuclear membrane, and then chromatin, another inner nuclear membrane, etc. So these structures can get very complicated. Actually, they're very simple in a way, and but it harkens back to the person we worked with, Howard Davis, really fine electron microscopist, who saw layering of chromatin, very highly reproducible layering of chromatin, in the chicken erythrocyte nucleus. The chicken erythrocyte, most mammals, we extrude the red cell nucleus. But in fish, in reptiles, in birds, the red cells have an inactive but nucleated red cell. So when the electron microscopy thin section studies were done by Howard, he showed that you get these multiple layers of heterochromatin, and on certain cross sections you see something that has been interpreted as 30 nanometer fibers. So we think that what's happening in the HL60 cells and I'm not going to show you a diagram of this. Let me show you one more from Howard. This is a tangential section. Tangential means that the knife, when you use a ultramicrotome, if this is the nuclear envelope here, you just do a grazing section of the top of the nucleus, and that's stained and examined, and you can see, so these would be nuclear pores here, and what you see is this concentric ring sometimes of what might be 30 nanometer fibers. So what Howard strongly argued is that the chromatin right near the nuclear envelope is very highly organized. We would guess highly organized by interaction with proteins of the internuclear membrane, such as lamin B receptor. Okay, so what we think is happening in the HL60 cells when they develop these multiple layers separated by membranes is that the increase in lamin B receptor is leading to a tremendous growth of the internuclear membrane, which is growing inward and sort of cleaving apart these sheets of chromatin, higher order structure, sort of like, you know, making a lasagna, if you want to think in culinary analogy, all right? And one last look before we leave these interesting structures, this picture that Ada showed. Here's a nuclear pore. Notice this fine layer right under the nuclear envelope. And what is, we still don't know the answer to this, and we're still puzzled, but we're so interested to think that maybe this, which seems to be maybe twice the thickness of that, represents 2 layers of chromatin fibers. Because remember, this is 2 inner membranes, and this only has 1 inner membrane. So maybe these 2 layers have interdigitated like our fingers might and give you a structure. Sheer speculation. No one is really studying this, and it's not really known what the answer is. Okay, now comes this is what we've done the last 2 years. Epichromatin and phosphatidylserine, a very interesting story, we believe. So this one is not actually showing that next slide I wanted to show. I guess we didn't get that on the stick. Doesn't matter. This is a story of 2 antibodies that we've been using, 2 mouse monoclonal antibodies. One of them is from an autoimmune mouse, and this is a situation we're collaborating with someone at Temple University in Philadelphia who's very interested in the mechanism of autoimmunity. And so there are some mice that spontaneously develop systemic lupus erythematosus. And you may or may not know, but in that condition, as in with many of the autoimmune diseases, anti-nucleosome antibodies, anti-DNA antibodies, anti-histone antibodies will appear. Well, in the same thing in these mice. And what he did was he took the marrow cells from he took the precursor cells and made hybridomas from the cells of these autoimmune mice that, by looking at their sera, had some positive anti-nuclear antibodies. So he generated a spectrum of monoclonal antibodies from these autoimmune mice, clonal and anti-nucleosomal. They were all tested to see if they reacted with chromatin by ELISA assays and by immunostaining assays. And we got a set of them from him. And one of them is an amazing one. It's called PL2-6. Doesn't matter, but you'll hear the name a couple of times. This is a confocal image of granulocyte nuclei. It's actually a projection of confocal images. So you're getting the impression that you're looking at the surface. These would be human blood neutrophils stained with this antibody, and then imaged by a confocal, and then a projection made through the slices. And you can see how beautiful this is staining the surface. And you can see how highly lobulated the granulocyte nuclei are. Now, when we use this same antibody and we look at confocal slices, and what we're going to show you is the confocal slices of HL60 cells. Okay, now this, I don't know how well it's showing up. Can we darken the light a little bit more? Unfortunately, a lot of the pictures I'm going to show you, can you darken the light a little bit more? Okay. Great. I know this does two things. It helps you see these pictures better, and it helps those of you who want to go to sleep go to sleep. Okay, so what this is a comparison of this antibody, PL2-6, with two other antinucleosomal monoclonal antibodies, also raised from lupus erythematosus mice, tested against HL60S4 cells and against U2OS cells. These are suspension cells, these are attached cells. And these are all the DAPI stained images, so you're just seeing DNA here. And in each case, so what we're showing you this particular interesting antibody, PL2-6, and it's compared against another one, and PL2-6, and it's compared against a different one. Actually, PL2-6 and PL2-7 are from the same mouse, and by ELISA, they have the same specificity, but they stain differently, which tells you that when you do an ELISA, you don't see everything. So let me first start with these. These antinucleosomal antibodies, and this is the DAPI equivalent, both of these are antinucleosomal. They stain the entire mitotic chromosome. This is a mitotic set of chromosomes. And this is a prophase. You're going to see more pictures like this, so I just want to get you, in tune with what we're going to show you. This is a prophase nucleus. You can see the DNA is beginning to condense. You'll see better pictures, believe me. And this is mitotic chromosome. So this antibody is staining just like the DAPI stains, okay? Then you go over PL2-6, this antibody that is so interesting to us, and it only stains the perimeter right near the nuclear envelope in an interphase nucleus, and it just, again, I promise you'll see better pictures. It stains the perimeter of the mitotic chromosomes. This is HL60 cells. If you go over to the U2OS cells, which are attached, again, here's this PL2-7, which nominally has the same specificity, but it's staining just like the DAPI. It's staining the mitotic chromosomes, and it's staining throughout an interphase nucleus. But when you look at the PL2-6, you can see it's only staining near the nuclear envelope in the interphase nucleus and the perimeter of the mitotic chromosome. So now you're going to see more and much easier to see. I'm sorry but I wanted you to see it. I wanted you to see what the other antibodies, how they are not doing what PL2-6 is. Okay, PL2 staining throughout the cell cycle. These are the U2OS cells. This is an interphase nucleus. The blue is DAPI, the DNA. You can see you get this nice, clean, sharp staining right around where the nuclear envelope would be. Now, early prophase, you can begin to see the chromatids are condensing here. And maybe there's still some nuclear envelope. It's probably beginning to fall apart here. The lamins are dissociating, and the nuclear envelope membranes are retreating with the ER towards the plasma membrane, which is out here somewhere. Okay, a little bit later in prophase, you can see now probably the nuclear envelope is completely gone here, but you can see just the outer surfaces of these condensing chromosomes show this epitope. Now you're into metaphase, and you can see again, it's only the outer surfaces exhibit this epitope. The inner chromosomes, condensed chromosomes, don't exhibit it. This is probably the plate, the metaphase plate, when the chromosomes have all congressed with one another. Again, the epitope is only on the outside. Now you're beginning to get early anaphase. The chromosomes are beginning to separate from one another, and you can see that again, the epitope is on the outside. And then in later anaphase, you're beginning to see a little bit of that epitope appearing here, and the trailing chromosomes. And then this is probably telophase, and you can see that the epitope is here, but there's a region here which is devoid of the epitope, and that's where the microtubules are inserting and where the centromeres are. And then this is probably around cytokinesis, and again you can see this, the epitope is here, but this region where the kinetochores are is still deficient, although later on in G1 it obviously has to surround the entire nucleus. Now, because this antibody only stains the outer surfaces, sort of like the skin, we've named this region of chromatin epichromatin, like epidermis. Okay, now I'm going to show you some movies, and I hope these come out all right. I'm going to show you some movies of this late prophase and then of a late telophase. So this is we made a movie from these are actually taken in a deconvolution microscope. So it's swinging back and forth, maybe plus and minus 45 degrees, something like that. So this would be the axis of rotation here. And what you can see now, unfortunately the colors changed. The green is the epichromatin, and the blue is still the condensing chromosomes. This, by the way, is a little micronucleus, which is commonly seen in tissue culture cells, and is probably from a chromosome that didn't make it into the proper spindle, and it's left out on its own. Okay, so here you can see nicely the condensing chromosomes, the chromatids, and the epichromatin. Now we'll turn off the blue so you don't see the DAPI anymore, and now you're going to just see the surface. And I find this one of the most remarkable pictures, because you can see it's just the surfaces that had been probably in contact with the nuclear envelope that are shining up with this epitope. Okay. See? All right, and this is a telophase, probably late telophase, where most of the chromosomes are beginning, a late anaphase, beginning telophase. So probably these chromosomes are beginning to sort of merge with one another, and there's a laggard here, a chromosome where the arms are still presumably somewhat attached to one another, and the chromatids are. And okay, so and this is an interphase nucleus. Okay, now we're going to turn off the DAPI staining, and you get a nice image of the surface staining of the epichromatin staining. Okay. Now I think we're going to jump. Oh, sorry, no, this is still with the PL2-6. And this is just to show you basically what you now know pretty well. That we're just comparing two nuclei. These are not nuclei anymore, two cells. This cell with this cell. This is an interphase cell with an interphase nucleus. You can see a typical DAPI image of an interphase nucleus. This is the epichromatin antibody staining on the periphery. This is LBR staining, okay? So they're very close to one another. They're very similar. There are some differences. All right, then in metaphase, or excuse me, this would be early anaphase. You can see the chromosomes pretty nicely. They're highly condensed with the DAPI stain. You can see, as we've told you, the epichromatin epitope is on the outer surface. Now you see lamin B receptor has retreated with all the ER towards the plasma membrane. It's left this area for the sake of the mitosis, and there is no nuclear membrane here. Okay. And then these are the merge images. Okay. All right, and then the ELKs. I told you I'd take you back to the ELKs. These are HL60 cells that have been treated with retinoic acid for 4 days. And then we do immunogold staining. So this is to convince you that they're both these lobulated nuclei with these ELKs, but these are two different cells. Okay. And if we focus on this region here where there are two elks next to one another that's enlarged, you can see the gold. This is called immunogold labeling. So the gold, the dark gold, is showing you where the PL2-6 antibody is. And you can see it's highlighting as best as one can with this kind of method the chromatin, which is only about 30-40 nanometers thick here. In this region here, which is just sort of a nuclear lobe, you can see it's on the outer surface here. And this is an interesting case where the nucleolus is right against the nuclear envelope, and it's separated from the nuclear envelope by one layer of heterochromatin, and this is also showing you the immunogold labeling. So this is bona fide, we believe it's bona fide, inactive heterochromatin, and it is clearly staining with this epitope, for this epitope. So this really does show you that the epichromatin at least is associated with heterochromatic regions. Whether it's associated with euchromatic regions is less clear because there's much less euchromatin or active chromatin or diffuse chromatin at the periphery of the nucleus. Okay, then finally, oh no, not finally, still a little bit more. And it's not finally because I have one more antibody after this. This is to show you that the epitope, the epichromatin epitope, is very highly conserved. This is a plant. This is actually these are tobacco cell nuclei, tobacco cells, an interphase nucleus. This is what you've seen before, metaphase configuration. This would be anaphase, and then this is probably telophase. Okay. Now, during the last year, we have, when we showed the previous data to a friend of ours who lives in Portland, Maine, we had been friendly for years with him. We showed him that previous data and told him the paper's coming out. He said, oh, I've seen that kind of picture for years. And indeed he had. He had been working with people and they had made an antibody directed against phosphatidylserine. What they do is they make liposomes that include phosphatidylserine and they immunize mice with it, and they wound up with monoclonal antibodies against phosphatidylserine. So for those of you like me who don't know, or in my case didn't know what phosphatidylserine is, I have a diagram, a little bit of chemistry here. This is this glycerol backbone with two fatty acid side chains. This is the phosphorus group that makes it a phosphatidyl. And then this is serine here. This is the side chain, the hydroxyl side chain of serine. It's coupled with the phosphate group. So what's interesting is that phosphatidylserine, which is in the membranes of the cell and is not a major component of cell membranes, is the major anionic lipid of cell membranes, and it's anionic because these two groups are basically neutralizing one another, and the phosphate is still highly charged. And it's known that phosphatidylserine in the plasma membrane faces the cytoplasm, except during apoptosis when it is exposed to the extracellular sites, and then it's detected as a dying apoptotic cell. Anyway, this phosphatidylserine interacts with basic proteins near the plasma membrane. Well, it turns out that when we use this antibody, an antibody directed against phosphatidylserine, we got, and I wouldn't be telling you this if we weren't astounded by it, we got an image that looks exactly like what we got with the anti-epichromatin antibody. So here, by now, you may be sick of it, but you can recognize an interphase nucleus with this very clean staining near the nuclear envelope, prophase with the beginning of with the breakdown of the nuclear envelope and the condensing of the chromatids, metaphase, anaphase, probably late anaphase, and then here somewhere telophase near cytokinesis. So this antibody gives an identical image to another antibody, which we showed you, which we call the anti-epichromatin antibody, which so far as we know, only has specificity for histone DNA complex. And this is nice control on NIH3T3 cells that Igor did, where we're comparing the two different antibodies. This 1H6 is the anti-phosphatidylserine antibody, and this is the anti-epichromatin antibody. So if we don't absorb the antibodies, this is the image we get. If we absorb the antibodies before the immunostaining with phosphatidylserine, it eliminates the binding, but it doesn't affect the PL2-6. If we try and absorb with phosphatidylcholine, there's no effect. And if we have a mixture of phosphatidylserine and phosphatidylcholine liposomes, it knocks out the binding of 1H6, but not of PL2-6. This is an ELISA experiment where we vary the antibody concentration, and the wells in the ELISA plate were coated with phosphatidylserine, and you can see that PL2-6 shows essentially no reaction with the phosphatidylserine on the ELISA plate, whereas 1H6 shows a very strong reaction. Okay. And then it turns out, just like the PL2-6 antibody, this 1H6 epitope is also highly conserved in evolution. So this is now a reaction with Drosophila KC cells. And there's another antibody being introduced here. You may not know about this one, but this is a histone modification antibody, which is very useful because it only reacts with mitotic chromosomes. It's called histone H3 serine 10 phosphorylation. So the S10 region of histone H3 is phosphorylated. And it only shows, it's called a mitotic marker, because if you apply it to cells, that it only reacts with mitotic cells. So you can clearly see this is probably late anaphase, and this is mitotic, closer to metaphase. And you can see that the 1H6 and the PL2-6 give nice reactions with interphase nuclei and with mitotic chromosomes. So again, even though these antibodies have very different specificity, they are giving pretty identical reactions by immunostaining. So this is my summary. Peripheral interphase heterochromatin and ELKs have a unique chromatin conformational epitope, which we call epichromatin, which co-localizes with phosphatidylserine. Secondly, these epitopes, the ones up there, persist throughout the cell cycle and are highly conserved in evolution. So we've come up with what we call an epichromatin hypothesis. And that hypothesis states that epichromatin, the surface chromatin, facilitates post-mitotic nuclear envelope reformation. That's a large phrase to describe this phenomenon that really is an amazing phenomenon that we think a lot about. Remember, let's say tissue culture cells or growing cells in your body might be dividing on a 24-hour cycle. So if they are, let's say, stem cells, they might be dividing every 24 hours. Out of that 24 hours, 1 hour is mitosis. Thank you very much. Yes, you can see I'm still here. I didn't walk out the room and turn on the recording. So that during that 1 hour period, the beautiful nuclear architecture has to be broken down. The nuclear envelope is disassembled. The ER go to the periphery. Who knows what else happens? Genes, they lose their organization to some extent. In that 1 hour, then the chromosomes are separated, and then the whole nuclear architecture has to be rebuilt in obviously less than an hour, because a lot of that hour is used for chromosome movement. So, how does the cell do it? Well, we don't know how the cell does it, but we're looking desperately for things that might provides some of the information to help the cell rebuild the nucleus. And so we think the epichromatin might play a role. That's our hypothesis. Now, another interesting thing is that lamin B receptor, which I ought to spend a lot of time talking about, remains embedded in the endoplasmic reticulum as it moves away from the dissolving nuclear envelope. And it is one of the first proteins, it's been shown by Roland Feusner some years ago, one of the first proteins to contact the nuclear envelope during reformation. So you can see where our ideas are headed, that there are factors embedded in the internuclear membrane, which is continuous with the ER, which furnish some information to help rebuilding the nuclear envelope, and there are factors in the chromatin which may furnish information for rebuilding the nuclear envelope. Well, finally, the nucleus. Well, I think the best way to think of the nucleus is this old ancient Indian parable. It's such a complicated structure, and most of us, and I like the way this is drawn, we've all got our white lab coats on. It's such a complicated structure that depending upon the techniques that we use, we come up with a different view of what the nucleus is. So this is not to depress the students. This is to show you that there's plenty left to do. You know, maybe Ada and I have said, well, it's awfully like a snake. But you've got the rest of the nucleus to unravel. So finally, we would accomplish nothing if it wasn't for our many collaborators. We've collaborated with people a lot of our collaborators in Heidelberg, in Berlin, in Munich, Portland. Igor was the one who showed us the 1H6. And then Lenny Schultz at Bar Harbor, who had the ichthyosis mouse, and Peter Gaines at UMass in Lowell, who's developed those wonderful EPRO cells. And this is a pretty picture of Heidelberg, the river, and the castle in Heidelberg, and the old bridge. So anyway, we've enjoyed much that we've done in large measure because of the wonderful collaborators that we've had. Thank you very much, and Ada and I will both be happy to answer questions.